just tell you very briefly about my career and then be open for questions. But I will give you a warning label as we start this lecture that what I'm going to really convince you at the end of the lecture is that it's a good idea to fail. You just have to decide what level you want to fail at. So, to start, I've titled my talk, Nevertheless, We Persist. And this is um, basically about my career over the last 30 years. Now, I started out like many people do in school, thinking about what I wanted to be, what I wanted to do. And when you go to school, sometimes it can be hard to make up your mind what you want to do, especially if, like me, you enjoy history and geography and you enjoy science and maths. And at the time that I was studying, it was a little bit difficult to figure out how to find a way forward. So my lecture is organized in 10 chapters, and these are just the titles of the chapters. But to start, if you look at that map of India, my father was in the railways, my mother was a teacher, is a teacher, and the black stars on the map are all the places where I lived before I was 18 years old. I wound up in some places more than once. So that meant that I went to 10 schools. And if you know how dislocating switching a school is, especially if you're moving between educational systems, you'll understand that this was really dislocating, and because you leave behind friends. And if you leave friends behind, how many sequences of friends can you really keep up with? You learn early that you have to adapt to new people, to establish systems, to establish curricula, new teachers, and it really can be quite challenging. So I learned early to deal with new situations. And when it came to making up my mind about what I wanted to do, I kind of varied between wanting to do medicine and wanting to do mathematics. And at that time, there were a very limited number of places where you could go for a good math degree, but lots of places where you could go for a good degree in medicine. And during that time also, my uncle, who was studying in Vellore, used to come back home every summer holiday and tell us about the wonderful time that he and his friends had. And they were constantly going on picnics. They were having visitors from all over the world. It just sounded like such great fun. And also, being from a family where there were only two daughters, Vellore had the added advantage of being about 2,000 kilometers away from my parents. So there were lots of factors that went into making the decision to study in Vellore. And that's a decision that I've never regretted, because after that, I've stayed in Vellore pretty much for the next 40 years. Even in college, it's very exciting to be a medical student, because the kinds of things that you learn, you do in biology, but if you have a passion for understanding systems, medicine allows you to explore it in many different ways, whether that is physiology, or it is the idea of public health, or if you're doing something like radiology, there's a lot of physics mixed in there. Doing biostatistics was great fun. Doing microbiology was fun. In ophthalmology, you get to explore a system that is small and enclosed, and where a deep understanding can really allow you to figure out what requires medicine, what requires surgery. So doing all of that, progressing through medicine, is obviously interesting. But even more interesting than that was the idea of college life. And in Vellore, because it is a small town, students were expected to make many of their own entertainments. So, as much as I was learning in medical college through the classes, I was also learning through the things that we were doing after class was over. So, whether it is learning to make friends, to get out with groups, I belonged to a social service group. We would go out and give measles vaccines, which were new at the time, to all the government schools in the area. We used to put on plays. I recognized very early that I had no talent at all for singing or dancing, but I was very good at arts and crafts. And so I was roped in to do props and costumes for all the plays that were done. And the picnics, of course, which my uncle had promised me, did happen on a regular basis. Some of them created interesting events for the exploration of infectious diseases, like the time when we decided to go to a place that was about 6 kilometers away. Being students, we cooked our own food and took it with us, and the entire bus developed diarrhea on the way back. Then it's a forensic exercise to figure out who had eaten what and what had caused that diarrhea. Maybe that was responsible for my subsequent career. When I finished my MBBS, I had to decide what I wanted to do an MD in. I really loved ophthalmology, the idea of a contained system in which you could function was amazing. I went to All India Institute, worked in ophthalmology there, realized that my family had an essential tremor. So being a surgeon was probably not the best idea for me, because as I got older, my hands would shake more. I thought about psychiatry because, again, this was a field that seemed fascinating, the idea of mental health and how people do not always have an understanding. How do when you're sick, in terms of a physical illness, you can have a good understanding that you're not well. But frequently in psychiatry, the people who are most affected have no understanding that they are actually unwell. So, how do you create an environment where you can treat them? So, that fascinated me. And then infectious diseases had always been fascinating, but infectious diseases was not a subject of study at the time. So I did an MD in microbiology, and after doing 3 years of bench research, which only consisted of samples, I realized that I did not really want to do this day in and day out. There is a certain level of clinical interpretation, but there wasn't the engagement with bigger problems that I wanted to address. And so I moved to research. And this here is a picture of the Christian Medical College, which has just grown and grown over years and is now a 27-bedded hospital. And at the back is a building in which I work. So at this time, the question really was, could I afford to do health research? Should I be doing health research in India? Is research a necessity or a luxury? The story we are often told is that if you want to provide healthcare to the country, then shouldn't you be the one that is working on actually treating patients? The idea being that good doctors and good nurses save lives immediately and not in the future. On the other hand, if you think about why research is essential, we are actually borrowing all of our treatments currently from what comes from Western textbooks. We don't define our own problems and our own questions enough. Even if you look at all the medicines that we take today for non communicable diseases or antibiotics, the dose that we give to people has been calculated based on studies that are on a 70 to 80 kilo man who is white. Now, will that translate to an appropriate treatment for a 45 kilo woman in India? It seems very unlikely, but we haven't done enough to determine what the right treatments are for that 45 kilo woman and all the other people who live in this country. Well, after I finished my MD and started to work on basically enteric pathogens, studying diarrhea, I realized that there wasn't enough going on in the country and I really needed to learn more. And I was fortunate to get a fellowship to go first to the UK and then to the US. And it was very empowering to discover that, while I was on the faculty by this time of CMC Vellore, I also had the capacity to do really well in the UK and the US. I went to the UK, I did my MRC path, subsequently got an FRC path, wound up working with the lady shown here, Mary Estes, who is probably the world's leading rotavirus researcher, and she's at the Baylor College of Medicine. So the choice was, I had options. Should I stay here or should I go back to India? And if I go back to India, what is it that I really can do? And one thing that was evident is that if you want to address problems, you need to think about which are the kinds of problems that you want to address. It's not like researchers in the UK and US don't do good research. They do really amazing things. We are learning from them all the time. But who does research for the bottom of the pyramid? Who does the research that really matters? And if you look at the field that I wound up in, studying diarrhea is probably the least glamorous field of research you could actually think of approaching. And as an academic researcher, what would I do? So, telling the government what is important, carrying out research projects that are needed to provide the evidence to government that this is important, to make new products, to make policies, and translate all the results of our research into practice in India. And for me, one of the most important things, make sure that we expand beyond our individual efforts to make teams that can carry out research and training so that there is sustainable research capacity in this country, that we expand what we are doing. Now, while all of this sounded really idealistic, I had gone from medicine in a hospital, microbiology in a hospital laboratory, and I came back saying, I'm going to work in public health. And the reality of public health is that it's very, very, very different from working in a hospital because you have to build trust with the community. You're going in there, you have to make a commitment that this is something that you are going to do for a really long period of time. And in public health and in my research project in particular, I found that the team that I was hoping would work with me um, had moved on to something else. So I had to rebuild everything, essentially starting from scratch, which meant that I had to learn to do a lot of things that I had never done before, whether that was collection of data or trying to understand what we could do with GIS mapping, filling in for doctors who didn't turn up for the clinic or field workers on holidays when absolutely essential samples have to be collected, and building a team. And because you have to recognize that once you start to do research in communities, there's no way that one person or even a small group can do everything that needs to be done. And when you try to do research, you know, convincing people that what you are doing is important is can be very difficult. especially if you don't speak the language, if you don't know people who are trusted in that community. Building those relationships takes time. And if you are going into community based research in order to test an intervention, you actually don't know what you're going to find. You're asking people to be guinea pigs to test what might be a new screening test, a new diagnostic, a new treatment, a new vaccine. But essentially, all of these require human experimentation. And you're asking people, let me try out this unknown thing with you. So, building the levels of trust that are required for the community to accept you and to work with you on this is not a simple task. So, we did a lot of things, and we made the commitment to the community. I started a clinic that was in the area. The clinic treats children in the area even if they are not part of our studies, and I just thought that it was my responsibility. And then the other thing is, when you do research projects, the research project will end, but your commitment to the community does not end at that time. We stuck with it. We were desperately writing projects when the last one is coming to an end. But I think ultimately what we learned was really important. We spent 15 years studying children that were born in the community. The work was on rotavirus, which I'll talk about later. But because we were working in the community, we also recognized that malnutrition, and particularly chronic malnutrition, were a serious problem. So if you look at these two children here, they both look happy and healthy, right? But the child who is dressed in black is about 7 centimeters shorter than he should be at the age of 4 years. The child in pink is 87 centimeters, which is the correct height for a 2-year-old child. They both look healthy, and this is an issue because the child in black is chronically malnourished. He is stunted, and stunting can lead to not only problems with physical development, but also with cognitive development. In 15 years, we learned that children in urban slums in southern India have 30% stunting. It was 65% when we started. It's come down now. The median IQ in the slums is 89 years, and children who are persistently stunted have much lower IQs than children who have never been stunted. So you can imagine what this means for the future economic potential of these children. Not only will they be shorter, and shorter also frequently means less strong, they will also have a lower IQ, and it's been estimated that a 4 point difference in IQ can lead to a 12% difference in your income when you are an adult. In addition to doing the work that we were doing with this community, we were also looking to see, could we find out more about rotavirus. And one of the things that we found while we were doing these studies was a finding that a very interesting strain that looked to us very much like a strain that had been found at the Indian Institute of Science was being seen in children in Vellore. And while this strain was being developed as a rotavirus vaccine candidate, in our studies, we could see absolutely no protection from this strain. Now, we published this information, and this had an unfortunate consequence in that the people who were developing this vaccine did not want to hear the news, which meant that for many years, I was not invited to any meetings that had anything to do with rotavirus, that there were many people who said many things that were not particularly nice about me. until they finally found that our results were actually correct and they dropped that strain from being developed as a vaccine. So, originally, two strains had started off as vaccine candidates. One of them was the one where we said this strain should not be developed as a vaccine candidate because we can see that it is not working. And for the other strain, because we did not see the strain, we didn't know whether it would work or not, and we had recommended that it continue to be tested. That strain actually went ahead, and in conjunction with the work that we were doing on rotavirus, we were actually able to build a rotavirus disease burden estimate for the country. Now, when you're thinking about an intervention, you have to tell policymakers why it matters. Why should we have this new treatment? Why should we have this new vaccine? And in order to be able to collect that information, you need to do a number of quite complicated studies that involve both the community and the hospital. So we did the studies, and essentially, what we found was that if you look at two children born in India, one of them will have a rotavirus diarrhea. One in eight children born in India will have to go to an outpatient clinic for rotavirus diarrhea. One in about 30 children will be hospitalized for rotavirus diarrhea. And at the time we did the study, one in every 350 children would die of a rotavirus gastroenteritis. So there were about 78,000 deaths that were happening every year because of rotavirus. So on one hand, we are collecting burden data saying that this is important. And on the other hand, we are telling the community that the vaccine candidate, at least one of the two vaccine candidates that you're working on, is not going to work. So the end result of these kinds of studies was that we were asked then to come and participate in the vaccine trial of the other candidate, which we did. And we worked with a group in Delhi, in Pune, and in Vellore. And in Delhi, Dr. M.K. Bhan, the former secretary of the Department of Biotechnology, who had identified the strain, was the one who asked us to come and join him in doing a phase 3 efficacy trial, which we did. And we found that the vaccine worked and the vaccine was licensed by the Indian regulatory authorities in 2014. So, from having said negative things showing that vaccines might not work, one vaccine candidate would not work at all, and also based on our other studies having predicted that a vaccine efficacy would be about 50%, we had at least one sort of success showing that this vaccine, the Delhi strain, was really working. This resulted in the indigenous rotavirus vaccine being introduced in 2016. And by 2019, we had the rotavirus vaccine covering the entire country. Now, we have two vaccines in the country. My group and I have worked on both of them. I've told you the Rotavac story, but we also worked on the other vaccine, which is made by Serum Institute of India. So there's something that India can really be very, very proud of. The fact that from start to finish, we developed the Rotavac vaccine in the country. But I brought up the story of the two rotavirus vaccine candidates to tell you that success is not always guaranteed. Both vaccine candidates had started off with a lot of promise, with everybody thinking that both of them were likely to work. And yet, when it came to doing really detailed epidemiological studies, really detailed laboratory studies, we found that one vaccine candidate was successful and the other one was not. And that's the way it goes in vaccine development. The rate of success is really quite low. Now, rotavirus vaccines worked 55% of the time in the children that we had done the phase 3 trial in. The same rotavirus vaccines work at 85 to 90% efficacy in children in Western countries. Why is that? And other investigators and I have been looking for the reasons why vaccines don't work very well. I divided the reasons into two. One is maybe because less virus is getting to the gut. These vaccines are live oral vaccines, which means that we expect the virus to go into the gut and start to multiply there. When the virus multiplies, then we will get an immune response which will protect against subsequent diarrhea. So if you give less virus, then maybe you will have a lower immune response. Or it could be that for some reason your body is not capable of making as good an antibody response. So we had a lot of reasons that we had proposed that could decrease the virus inoculum. Maybe the mother is transferring antibodies to the child, and those antibodies bind to the virus and stop it from getting in. So then you don't get replication and you don't make a good immune response. Maybe stomach acid is destroying the virus that we are giving in the vaccine, or other factors that affect immune responses, giving other vaccines, damaged guts, micronutrient deficiencies, host genetics, all of these could potentially affect antibody responses. So for each of these, we have actually done a clinical trial to try and figure out if we block some of these factors that might affect immune responses, can we improve the response to vaccines? So far, I have had one tiny little success, which is when you give zinc and probiotics, you can get a slightly better immune response to the rotavirus vaccine, but not enough for it to become a public health intervention. So everything that you see here checked off is one failed study of ours. The two crosses, stomach acid and host genetics, are not studies that we have done as yet. So with all of that, 30 years of working, we've done a lot of things. We've survived a lot of failures, a lot of challenges, and my group has grown a lot during this time. But more important than just the people who work with me are the thought partners and the friends that I have. The people to whom I can talk about the work, come up with new ideas, discuss with them, and think about what is it that we are going to do next. It is very lonely to do research by yourself, but once you start to do it as a group, it just becomes more and more fascinating and more interesting. So right now I've decided that I'm going to switch gears. I'm going to be very ambitious, and there are two directions I'm planning to take. One is to focus on another public health problem that we have, which is why are birth outcomes in India really poor. If you look here, the figures that are shown for India, for Assam, and for Karimganj district clearly illustrate the fact that maternal mortality, or women who die in childbirth, and infant mortality in Karimganj are more than double what is seen on the average in India. So I am already starting to work there. And what we are trying to do is risk stratification. So when you think about a woman in pregnancy, usually what happens is if we can afford it, we all go to the highest end hospitals that we can find. This means that care is not being appropriately channeled. Usually, if you have no risk factors, it should be possible to manage pregnancy and childbirth at a lower level facility. And it should be only high risk patients who go to the referral hospital type of care if they have any kinds of risk factors that indicate that they need it. So, what we are planning to do, we started on the baseline at the moment, is to try and figure out what kind of high level monitoring, what kinds of readouts do we need from clinical and biological and imaging data that will allow us to do risk stratification as early as possible so that we can channel women in the right kinds to the right kinds of facilities. And building a platform like this which collects high dimensional data on pregnancies in a cohort of defined populations in a remote part of the country, this will also give us a platform where we can subsequently test interventions that could be focused on either the mother or on the fetus. I'm also thinking about another challenge for vaccines. In Hyderabad, Bharat Biotech developed a new vaccine called a typhoid conjugate vaccine against typhoid fever. Now, typhoid is a problem of India. And Bharat Biotech did phase one, phase two safety immunogenicity studies in India and got the vaccine licensed. But when this vaccine had to be used in other parts of the World Health Organization asked, but where is the efficacy data to show us that the vaccine really prevents disease? And there was no data available. So Bharat Biotech went to the University of Oxford, which has just had a vaccine license today, and with the same group that has done the SARS-CoV-2 vaccine trials, they did a typhoid challenge study, essentially where people were given the vaccine and then challenged with the bacteria to see whether the vaccine could protect them against typhoid infections. And they found that the vaccine had 85% efficacy, and then this vaccine started to be used in countries in addition to India. So it's been used in Nepal, Bangladesh, Malawi, Pakistan, Zimbabwe after its WHO pre-qualification. But my question really is, why did they need to go to Oxford to do the studies? Why could those studies not have been done in India? These are very safe ways of doing clinical research, and we should think about it. Unfortunately, we have not enough thought and a very active media which comes down in different ways, telling us that this kind of guinea pig, treating our population as guinea pigs is inappropriate and unethical. And my question back is to say, well, if we want new drugs and we want new vaccines, where are they going to come from? So, in fact, the UK is planning now to do challenge trials with SARS-CoV-2 as well. And this will allow for the next generation of vaccines to be tested uh, and down selected. So, what have I learned in 30 years? I've learned that it's very important to have a strong foundation. It's very important to make time for family and friends and never be afraid to ask for help. This is particularly for women in the room. Family and friends count just so much for all of us, but with their support, there's really a lot that we can do. As a scientist, be curious, ask questions, and find the answers. We can find the answers even if they are not what they expected. Or if we can't develop the tools, if they don't exist, then we will find out that it can't be done quite that way. And we have to figure out that there must be other routes to addressing these issues. I think believing that you can and will solve problems is very important, but it's very important also to stay with the problem long enough, not give up if something does not work and switch to something else. And it's very important to have the right inputs and get the right help if you need it. The biggest advantage of working in India is that no matter what problem you choose to address, whether that is in physics or chemistry or biology or medicine or anything, you can make a difference. There's such a need for good science that I think that we will never reach the limits of what human endeavor can achieve. And if this past year has shown us anything, it has shown us that science and technology can and will solve problems. So thank you very much for your attention, and I'd be happy to take questions now.
Thank you so much, Professor Kang. So there has been like a storm of questions coming in, and I will combine some of the questions because they run on a similar theme. And I'll start with the questions which are on COVID vaccine. So people want to know, how are these COVID vaccines being made? Um, why do vaccines have side effects, if they do have, and if they do, then why do they have? And also, when now that the second, I mean, there's a new strain of coronavirus that's prevalent, the vaccine development that has been ongoing, will that be sufficient for the new variant?
So I'm sure that in school everybody's read about Edward Jenner, right? What did he do? He took a cowpox virus, he put it into the arm of a person and made a vaccine that was protective against smallpox. Now, to take a living organism, to take proteins from a living organism, or in some very rare cases, polysaccharides that belong to a living organism or a virus, if we can call that living, then that allows us to give that entire organism or a component of that organism and induce an immune response from the host. That's the way that vaccines were made for 200 years. So give the whole organism, live or dead, or give part of the organism. Now we've got new vaccines where you don't give the protein or the polysaccharide, you send in the message for how to make that protein. And in the case of mRNA vaccines, DNA vaccines, and viral vectored vaccines, all we are doing is sending in the message. Then that message gets inside the host cell. The host cell then makes the protein, releases it, and the body responds to it. So, the advantage of the newer technologies is that actually you are giving fewer components in order to induce an immune response. And each time you give a foreign component, one of the worries is, are there going to be side effects to that? So the less you give, the less you have to worry about. So if we think about the new technologies, they're actually being designed that way to be safer. Now, when we test vaccines in vaccine trials, if you were to ask me, is a vaccine 100% safe? I will say no vaccine is 100% safe. No drug is 100% safe either. You can overdose on vitamins, right? So, aspirin, everything is unsafe. It's a question of how we use it. Now, in testing of vaccines, there's a difference between vaccines and drugs because drugs are given to people who are sick. Vaccines are given to healthy people. So, what we want from vaccines is a very high safety bar. And that's why when we test vaccines, in every phase of vaccine testing, as well as after vaccines are licensed and when they start to be used, there is safety surveillance. Monitoring of safety becomes very important. Now, why is that important? When you test vaccines, you test them in phase one in less than 100 people, in phase two in less than 1,000 people, in phase three in less than 100,000 people. Our current trials for SARS-CoV-2 are 30, 40,000 people. Now, if you have a side effect that happens 1 in 100 people, will you find it? Yes. Will you find it if a side effect happens 1 in 1,000 people? Yes, because you're immunizing 15, 20, 30,000 people in these larger trials. Half the people are getting vaccine, half are not. What if a side effect happens 1 in 10,000 people, 1 in 100,000 people, 1 in 1 million people? You won't be able to detect it in a vaccine trial, any vaccine trial, no matter how long it goes. So that's why safety monitoring after the vaccine is licensed is also very important. And we have the National AEFI surveillance system in India. AEFI stands for Adverse Events Following Immunization, which has been running for more than 10 years and is being strengthened at the moment to look for signals that might indicate that there are safety effects with the vaccine. What have we heard so far about safety of these vaccines? We've heard about the transverse myelitis, which is a neurological condition, or let's take all neurological conditions. Totally, we've heard about 4 such conditions around the world, and we've given 100,000 plus doses of vaccine. Now, is there a relationship? Right now, we can't figure it out because it's too rare. It's something that we need to monitor. We've heard about 6 cases of anaphylaxis or allergic reactions. Here, people are beginning to think that it might be the polyethylene glycol, a chemical that is present in the mRNA vaccines and helps to stabilize the nanoparticle that makes up the vaccine. This will be investigated. PEG is very commonly found in all kinds of products. We eat it, we drink it, we apply it in cosmetics. So it's there in a lot of things. Is it causing the allergic reactions? We don't know yet. It's a hypothesis. Now, if we look at what else has been, I was actually fascinated by the idea that 2 people who had Botox injections also seem to have an allergic reaction. Again, these signals, once they are detected, we need to investigate in much greater detail to figure out what the safety issues are so that appropriate warnings can be given to people who might be at higher risk of a safety event. But the one thing that I think is important to understand is that in the development of the vaccines that we are seeing for SARS-CoV-2 now, there has been no shortcuts. There have been no, let's not do this to advance vaccines. What has been changed and why we've been able to do this so quickly is 2 reasons. One is usually when we do these studies, we do them in sequence. I told you the rotavirus vaccine, one failed, one succeeded. Usually the chances of success for a vaccine are about 10%. So usually if you're doing animal study, then phase one, then phase two, then phase three, you'll be stopping after each stage and then starting the next one. You'll be waiting, submitting results, getting approval, getting the money, doing the study. What has been eliminated are all those in between phases. And the studies have been made much bigger than we really need in order to get results faster. So really, these are some of the biggest trials that have been done. These are some of the most robust trials that have been done. So when vaccines come, we can be confident in them.
So somebody just asked, the minister in Haryana who was given the vaccine still tested positive for the virus. How did that happen?
So it takes time for us to make an immune response, whether that is infection or vaccination. What it requires is for the organism to get in, or for the protein of the organism to which we are going to respond to get in or to be produced. And then it takes the immune system some time to recognize, oh, this is foreign, I need to do something about it. And that time is the time taken to induce a protective immune response. Mr. Vidge was supposed to get 2 doses of the vaccine. He got only one, and he got this about 2 weeks after he had gotten his first dose of the vaccine. So he should not have expected to have been protected at 2 weeks after the first dose. This is not unusual. You should start to count protection after you have received two doses, and not just the day after you get it, but 10 days after you get the second dose of the vaccine.
So, somebody wants to know what is the difference between a vaccine and an antibiotic?
Vaccines are ways to prevent infection. So, what you do is you give the vaccine before you expect the person to be exposed and get sick. Any kind of infection requires for the infectious agent to come into the body. Now, with a vaccine, we mimic the natural infection, and induce an immune response which protects us without actually allowing the person to get sick. So, for example, if you take a look at measles, everybody will get measles if the population is not vaccinated. Once you recover from the measles, you will be protected, and most likely protected for a long time. But that first infection might kill you. So what you want to do is to make sure that you protect the person from measles before they get it. And you do that by giving them a vaccine, which consists of the measles virus, but it only induces an immune response and does not make people sick. An antibiotic, on the other hand, is not a prophylactic or a preventive, it is a treatment. Once a person has already had the infection, you want to send in a chemical that stops that particular infectious agent from multiplying and producing the effects that make a person sick. So, antibiotics are used for treatment, vaccines are used for prevention.
So, somebody also asked a question that if somebody has coronavirus, will they recover after taking the vaccine? So, the answer is no. You should take it before you get the infection in order to avoid it. Okay, now there are a lot of questions around mutations and how does a microbe multiply. So, I'll again combine some of these questions. How exactly does a microbe multiply? What are mutations, and do mutations change the outcomes of a vaccination development or the strategies around it? Why is there no vaccine against common cold or medicine against common cold?
Sure. So, how does a microbe multiply? Well, if you are, some kinds of microorganisms can multiply on their own. So, for example, bacteria or certain kinds of parasites can function by taking in nutrients and dividing into, well, bacteria undergo fission, so can divide by producing from one organism two daughter organisms, right? Others can have one organism that produces multiple other organisms. Viruses don't multiply like that. Viruses are incapable of replicating on their own. And some parasites are like that as well. Viruses and some parasites need a host, which means some other living organism which has to help them to multiply. So in viruses, essentially a virus consists of genetic material which is wrapped up in a bag of proteins. That bag of proteins gets inside the body, The bag of proteins either goes in entirely or partially, but the goal of that bag of proteins is to deliver its genetic material inside a host cell. Once that genetic material is inside, depending on what kind of material it is, whether it's RNA, DNA, single stranded, double stranded, what it will essentially do is instruct the host cell to make copies of itself. Viruses can come out without destroying the host cell, or destroy the host cell to come out. So when viruses come out is when we can make immune responses against them. However, when we give a vaccine, what we want to try and do is prevent the virus from ever getting inside the cell. And that's the goal of prevention of infection and prevention of replication of viruses. So that's how organisms multiply. And you're going to have to repeat your other questions for me, Somdat.
So, if there are
When,
mutations, does
yeah,
it change the outcomes of vaccine strategies?
yeah. So it depends on the mutations. So
Maybe you can also explain what is a mutation
yeah.
first. Yeah.
So um, everybody knows American spelling and English spelling, right? So in American spelling, when you say pediatric, you have only PED, whereas if it's English spelling, it's PAED, right? Now, the meaning doesn't change whether you use American or English spelling. So if you look at mutations, you can think of them as spelling errors which make the meaning change, or spelling errors that don't make the meaning change. And if a spelling error does not make the meaning change, that is essentially irrelevant to us. It happens all the time. It happens more frequently if you have an RNA virus than a DNA virus. But if the spelling error makes the meaning change, the purpose of the genetic material is to make proteins. And proteins have a structure which is in all three dimensions. Some proteins are stiff, some proteins are floppy, some proteins will change their shape when they stick to something else. So what we are looking for in a mutation is, does the shape of the protein change at all? Because this shape is going to be very important for vaccines to work. If protein shape changes, that means that the antibody that is directed against a part of that protein will not be able to stick to that part. So when we look at mutations, we are looking to see whether these mutations might be relevant for a number of different reasons. One is the ability of the virus to stick to its host. The other is the ability of the virus, or that part of the protein of the virus, to be bound by antibodies, to have the antibodies stick. Now, what we are seeing in terms of this new variant that everybody is talking about is that this virus has the ability to stick somewhat better to the host cell receptor that is on the surface. So if the virus sticks better, its ability to get inside the cell becomes that much easier. Now, does that mean that vaccines won't work? We don't know that yet. Right now, all we know is that the virus is able to get in better, replicates at a somewhat higher level. So there's more virus around and it spreads a little bit more easily. Now, when we think of a protein, and as I said, there can be a stiff structure, it can be a floppy structure. The SARS-CoV-2 has a protein which is its spike protein on the surface, which is the protein that is used for attachment. And that protein actually is in different shapes before it sticks to the host receptor and after it sticks to the host receptor. Now, if you want to make a vaccine, you want a vaccine that will induce antibodies that are in the shape of the virus before it sticks to the host, because you want to block its ability to stick to the host cell. So right now, what we know is that there are many, many antibodies that are made to the spike protein. In the UK variant, of all of the antibodies that are made against the spike protein, there are one, maybe two, where the antibodies might not bind as well. But there are many others that will bind well. So we have no reason at this time to think that any vaccine that has already been made is not going to work on the UK variant. So that's something that we should feel reassured about. It's also very important that CCMB and other laboratories are now going to do on a regular basis genetic sequencing, so that if there are new variants that come around, they will be identified quickly and can be tracked through the population. Three sources of data are really important. We are hearing a lot about the UK variant. That's only based on sequence data. Sequence data is important, but we need more than sequence data. We need data that comes from epidemiological and clinical data. How are the patients who are infected doing? How are the populations in which this virus is circulating doing? And the third component, in addition to the genetic and clinical and epi data that we need, is the laboratory understanding of the mechanisms by which the virus and its interactions with the host are affected by the mutations. So without all three, we can't have a full picture. Just focusing on the sequencing doesn't really make sense. It's good to track, obviously, but insufficient to understand everything about the virus.
Okay, before I bring more questions from the question answer box, there are few students who have been waiting to ask you questions directly. So I'll unmute them one after the other. So, Satvik, I'll go first with you. If you're around, please ask your question. You're unmuted now. Okay. If Satvik is not there, Vanshika, I will unmute you now.
Good afternoon, ma'am.
Good afternoon, Manchika.
Ma'am, as we are hearing, various kinds of variations are coming in coronavirus in UK. What is the difference between the present virus and what is the variations in UK, ma'am? Like in present situation, we if we touch surfaces which is already like touched by the person who is affected, and if he speaks, we if the droplets fall on our body, then also we are affected now. So
Yeah.
what is the difference for both variations?
So, Vanchika, one of the most important things to understand about any infectious disease is that you need a certain number of copies of that infectious agent to get infected and get sick. So, for example, if you have to get cholera, then you have to have at least 1 million cholera bacteria to make you sick. But if you are looking at Shigella, which causes dysentery, then you need only 1,000 Shigella bacteria to give you dysentery. So, with this particular virus, we don't know what the infectious dose is, how many particles of this virus we need in order to be able to infect another person. We think that it's relatively small, and obviously, if a person is putting out 10,000 particles versus 100,000 particles, the person who puts out more particles is going to be more infectious. What we are seeing with this new UK variant is that it is allowing people to produce somewhat more virus than the older variants that we have circulating in the country. So we are seeing an increased level of spread. Probably because there is more of the virus that is being shed by people who are infected. Okay. Now, in terms of touching surfaces and people breathing and all of that, the most important things,
Hello.
as you know well, are masking and physical distancing, with the idea that you reduce the number of viruses to which you are exposed. Okay. So there's nothing that we really need to change about what we are doing. The same practices that we are following now to prevent infection are the practices that we need to continue to follow. And in terms of the variants and what we do about them, it's really around testing, tracing, and isolating, which is what the public health authorities are already doing with the help of institutions like CCMB.
Thank you, ma'am.
Sakita, I will unmute you now. You can ask your question if you're around.
Hello.
Good afternoon, ma'am.
Hi, Sakita.
Yeah. So my question is that as you said while somebody was asking a doubt, you said not all the vaccines are 100% safe.
That's right.
Yeah. So, but while I was watching news one day, the news was telling that Moderna vaccine declared that their vaccine was 100% safe. So, is that true or not?
So, what I mean by 100% safe is that no vaccine can guarantee that it will have no side effects. And even the Moderna vaccine causes pain on injection, causes fever, causes fatigue. It does not last for very long, 1 or 2 days at most. But we don't know yet whether there are any very rare side effects of the Moderna vaccine. So if something happens 1 in 100,000 people, we will not be able to say that from Moderna's trial, which only included 30,000 people. So take another example. We have polio vaccine. We've been using polio vaccine for a very, very, very long time, right?
Yes, ma'am. Yes.
But polio vaccine, the oral polio vaccine can revert to neurovirulence and can cause polio. This happens 1 in 1 million children. So no trial would have told us that this was going to happen. But once you start to use the vaccines, you will find out that these have side effects. Now, the question is the side effect so big that we should not take the vaccine, right? So you have to balance the benefits and the risks in order to make a decision about to use the vaccine. And where all these vaccines are concerned, even with the very low level of risk to young people, it's still the benefit risk ratio is very, very, very heavily weighted in favor of benefit. So if vaccines are available, everyone should take them. The risks of these vaccines, so far, we have not had risks that have been unequivocally associated with the vaccine. That's what Moderna says, beyond the pain, redness, fever, etc. There are no serious side effects that have been associated with the Moderna or the Pfizer or the AstraZeneca vaccine. That does not mean that once we use the vaccine in millions of people, we will find no association. So, being a scientist, I will never guarantee that any drug or any vaccine is 100% safe. But very important to consider that we must look not only at risk, but also at benefit.
Okay, ma'am. Thank you.
Lavanya, I will unmute you now. Lavanya, if you're around, you can ask your question. Okay, we'll go to Mehul. Mehul, are you around?
Yes, ma'am. Good evening, ma'am. Ma'am, my question is that where if a person is affected with the coronavirus and he gets cured before he has got a dose, and after getting a dose of a vaccine, is there any chances that he can be affected with the virus?
So, with most mucosal infections caused by viruses, we see reinfections. But usually, when you have a reinfection, that infection will be milder or asymptomatic. Now, in terms of if a person has been infected with a virus, will they have some level of immunity following infection? Yes, that is very likely. Should they receive a vaccine if they have already been infected before? Well, we know from people who have been infected before that the level of antibodies that they make is quite variable. What I had said earlier about infectious doses, there is a difference in the amount of virus that infects you that may affect how much of an immune response you make. Your own host response might affect the level to which the virus is replicating. So, giving a vaccine which has a defined amount of antigen, which is being made to induce a good immune response, is useful even in people who have previously been infected. And once infected, does not mean that you will never be infected again. It does mean that you likely have protection against illness. Being infected does not mean sick. A lot of people can be infected without ever getting sick. But if you've been sick once, then chances are that you will be less likely to be sick when you have an infection the second time.
Ma'am,
Shri-
Okay,
yeah.
ma'am. Thank you.
ma'am,
Shrikar, I will unmute you now.
excuse me, ma'am. I'm so sorry. There was some problem with my mic, ma'am. May I ask my question?
Okay, go ahead.
Thank you so much, ma'am. Good afternoon, ma'am. Ma'am, my question is regarding the rotavirus, ma'am. Ma'am, you had mentioned about it, ma'am. So, how infectious is rotavirus, ma'am? And where is it found, ma'am? And what is the source of the virus, ma'am? And does it still exist in India, ma'am?
I don't think we are going to eradicate rotavirus anytime soon. So, rotaviruses are viruses that are found in animals, in birds, and in humans. They usually cause diarrhea in the young of any species. So, young horses can have rotavirus diarrhea, young children have rotavirus diarrhea. It mainly infects the gut. And the kinds of vaccines that we are using are intended to reduce disease. They don't necessarily reduce infection, but they do reduce disease. And rotavirus, because it's found in both animals and humans, is not going to go anywhere. I think we are going to continue to have it circulating. The way that children acquire infections, children and adults acquire infections with rotavirus, is really through contact and fecal-oral transmission. So it's a gut virus. You can get it from eating quite rarely, but mostly you get it from person to person contact. So very common for parents of young children to have uh, get a rotavirus infection, but otherwise usually it's only seen in children.
Thank you so much, ma'am.
Excuse me,
Yeah, Shrikar, go ahead.
ma'am. My doubt is uh, during the last year, many misconceptions started circulating about vaccines. Many misconceptions were there, such as one misconception was saying that it causes autism. Another misconception was it was extremely dangerous and its side effects had caused death in some people. So these misconceptions are spreading very rapidly, ma'am. And do you think that these misconceptions have any basis, have any facts to back them up?
I think scientists are not very good communicators, and people who are anti science are much better communicators than us. So, what has been happening and what you are highlighting is a very, very important problem, which is only going to increase with time. The study, the basis of saying that vaccines are associated with autism was from a study of children with autism where it was claimed that they had received a measles vaccine before they manifested with the symptoms of autism. The paper was published in the Lancet. The paper has been retracted. The author who wrote that paper has been struck off the medical register for unethical practices. And yet this story persists. So it's also been shown that this is a feature of human communication. We remember bad news and we don't necessarily remember positive messaging. So the associations of harm, the associations of dangerous events is something that people remember. And this is what the anti-vaccine lobby is using to its advantage and communicating very well. Autism is one issue. The other issues of serious side effects that you have highlighted have also been shown. But if a vaccine killed people, why would a regulator license that vaccine? The responsibility of a regulator is to ensure first quality, second safety, third efficacy. So the quality comes from the manufacturing process where they assess the vaccine as it is made, every batch of vaccine. The safety comes first in clinical trials. If a vaccine has side effects, that vaccine is not going to be licensed by a regulator, even if it is a good vaccine. It all comes down to, as I said before, the benefit risk ratio. The benefit has to be much, 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 much greater than the risk. The risk has to be very, very, very small for a regulator to allow for a product that is going to be given to healthy people. Remember that this is not a cancer drug. If it was a cancer drug, we would accept side effects because you know that the person with cancer might die without this drug. So, hair falling out, loss of weight, skin color changing, all that we've seen with people who get chemotherapy, those side effects are acceptable because that drug treats a disease that might kill the person otherwise. When you're thinking about something that needs to be given to healthy people, the bar for safety is very, very, very high. So, no dangerous vaccines are going to be allowed.
So I'm going to unmute the last person who happens to be Tejas simply because his name comes up here. But after that, we will close the session. But yeah. Tejas, you can go ahead.
Hello everyone. Ma'am, my doubt is now it's a fact. I've heard it many times. It's that most of our DNA structure is retrovirus. Now, a child like me would assume that okay, maybe viruses have their own triggers. If we compare coronavirus, it just comes into our body and within 2 days of introduction, it starts working against our body. Whereas some viruses, they just come into the body and they become a part of our DNA. Ma'am, so how far is my assumption correct, first of all? And second doubt related to that is there some way to stop these viruses from being triggered if that trigger assumption of mine is correct?
So, our genome is a well-known fact, but retroviruses have a special feature that allows for that integration. They have a reverse transcriptase that allows them to be read, which SARS coronavirus 2 does not. SARS-CoV-2 is an RNA virus, but does not have a reverse transcriptase. And the way that it exerts its effect is by multiplying and multiplying not in the nucleus of the cell, but in the cytoplasm. So what it's doing is getting in and hijacking the protein making machinery of the cell in order to be able to replicate. So even though retroviruses are RNA viruses and coronaviruses are RNA viruses, the two are very, very different in the ways that they interact with their host cells. So certainly for this virus, you don't need to worry that the virus itself or any vaccines against the virus are going to integrate with the host genome. Retroviruses and the functions of what ancient retroviruses in our genome are doing are something that is more aligned to the work that CCMB does than me. So I'll leave it to them to answer the questions while you're spending time with them. I just wanted to address one question which wasn't addressed before, which was why don't we have vaccines against common cold viruses and the difficulties of making vaccines overall. So if you look at the coronaviruses that cause common colds, there are four of them. There were originally five that were discovered, four that we know of at the moment, that circulate in human populations quite a while. And you have to think about, what do I want to make a vaccine to? Do I want to make a vaccine to a disease that is a serious threat or something that is an inconvenience? So usually when vaccine researchers are looking at what should I make a vaccine for, they will choose a disease that has a significant burden, which means the disease has to affect a lot of people, which of course common cold viruses do, but also the disease has to have some level of severity that people would want to invest money in preventing so that the company can recover the money that it invests in making those vaccines. So while we have many kinds of infections, we don't make a vaccine for every infection. We make vaccines for serious infections. So really, we haven't focused on making vaccines for the common cold coronaviruses because those are not serious or life-threatening in any way. But for SARS-1, we tried and then the disease went away. We are working on vaccines for MERS coronavirus, and obviously we now have vaccines on SARS-CoV-2, which will allow it, which will make it a lot easier for us to make vaccines against other coronaviruses in the future.
So before we wrap the session, I'll go away from the viruses and ask one final question, which was around your career. So somebody wants to know, uh, yeah, while you were younger, you were interested both in mathematics and medical sciences, and you picked one. I mean, you went ahead with medical sciences. So today, do you feel that you would have also wanted to pursue mathematics?
So in fact, most of my work in public health is quantitative, because if I can't measure something, I can't do anything about it. So in all the work that we do in the testing of interventions, measurement is critical. Now that's not fancy mathematics, but it is careful quantitation. And then the analytic techniques that we use, now I'm working with mathematical modelers, which I find absolutely fascinating. So I guess I came back to marrying my interests together.
So maybe there is no distinction between subjects so much as we think, as our textbooks make us think.
Absolutely.
So, though there are many, many questions that we have not been able to address, maybe I can tell the audience today that Professor Kang is also available on Twitter. And if you want to continue some of her discussions, you can post things there. And if she is free and when she is free, she'll probably take a look at them. Uh, Professor Kang, thank you so very much for this engaging discussion today. I'm very sure that there are many people who are thrilled with the kind of work that you have done. And possibly we have inspired the next generation of scientists through this discussion today. Thank you so much. Uh,
And to Advait Sri Kumar, yes, I felt depressed.
oh.
But that's par for the course. You have to get used to the idea. So the basic thing is, if you are not failing and you're not challenging yourself enough, right? So you have to pick the big problems and then you have to stick to them. And yes, you will feel depressed. Yes, there will be times when you will feel like you've taken on too much. You will feel like nobody is supporting you. But when you finally get to the point where you find an answer, positive or negative, and you know that you can trust that answer, that is the making of a scientist.
Absolutely. And making of thinkers as well. So thank you so much.